Hello, everyone. Welcome to today's SONA webinar series. I'm Mahmoud a research fellow at the University of Sussex in England. Regeneration. And as you know, the purpose of the SONA webinar series is to make neuroscience more accessible to all so that we can get experts from around the world to talk to kind of researchers, especially in Africa, but also in other parts of the world. And at the same time, also to help in creating mentorship opportunities. And today I'm very, very excited to say that we have Professor Giovanna Mallucci. And I'm going to give you a brief background about Giovanna, but to be honest with you, if I were to spend the next 10, 15 minutes, I could go on and on talking about her kind of accomplishment. So this is just going to be a brief summary. Giovanna is a professor of clinical neurosciences and an associate director of the UK Dementia Research Institute at the University of Cambridge. Her undergraduate degrees were in physiological sciences and medicine from the University of Oxford, with clinical training at University College London. She obtained her PhD from London University, and her lab is pioneering work interventions targeting common pathways for treatment of dementia. Her work on the role of the UPR in neurodegenerative diseases and its pharmacological manipulation was hailed as turning point in search for medicines to control and prevent Alzheimer's disease. And this was reported in the BBC. And also, her work was, in 2013 and 2014, her work was reported as the Society Success Stories for the MRC. And she has received numerous national and international awards for her work, including the Scientific America 50 Award for Leadership in Research as one of the top 50 scientific innovators worldwide. And she has also given many plenary and keynote talks at international conferences. She is an ERC Consolidator Fellow. In 2017, she was elected Fellow of the Academy of Medical Sciences. She's also honorary consultant neurologist at Addenbrooke's Hospital, specializing in dementia. And I was very, very fortunate to have her as my external examiner 3 years ago to examine my PhD. So Giovanna, we are really, really excited to have you today, and the stage is all yours.
Thank you very much, Mahmoud. Thank you, everybody, for listening. I think I'm going to disappear now, yeah?
Okay.
Get full screen. Yeah. I'm going to talk you through this lecture, which is really a story about how we approach the problem of neurodegeneration, and it's how difficult it is to treat in two different ways, and mainly to open up the idea that there are all sorts of ways to target understanding disease and treating it. And then at the end, I'm looking forward to speaking to some of you in a question and answer session. But I've called it Mechanisms to Medicines because this underlines our approach. So just to start, as a general introduction, this is a group of disorders, neurodegenerative diseases, which go from the very common to the very rare. And certainly in the UK and most of the developed world, Alzheimer's is a very common disease, as is Parkinson's, but these become rarer. But these are all diseases of old age and the elderly. So as the world is aging, which is happening globally, we are facing a massive pandemic of untreatable degenerative disorders in the older population. And these are going to be the commonest cause of death worldwide, but very soon. And at the moment, we have no treatments for them at all, no disease-modifying treatments. So in a world where we can treat HIV, we can treat cancer, we can transplant organs, we can do nothing at all to slow down any of these diseases, which are increasingly common. So there's been lots and lots of publicity about attempts to treat them, because all of these disorders have associated specific proteins associated with them. So I'll tell you a bit more about that in a minute. But the basic thing that you need to really think about with these diseases is that these are neurons or brain cells, which each disorder, whichever one you choose, each one has a pattern of neurodegeneration, brain cell loss. And this is particular for each disorder, but it's a common feature. The other important thing is that between each brain cell, there are millions of synapses, and the synapses are these connections, and I'll talk more about these later. But these are where memory is stored, where information is passed from neuron to neuron, but also they are essential for keeping neurons or brain cells alive. So when you lose enough synapses, you lose neurons. And I'm spending some time on this slide because I want you to realize that these are entities that come and go continuously. It's something that we're continuously making synapses. Every time we're learning, every time we're sleeping, every time we're doing a new task, during development, synapses are being formed. So this is an element of the brain cell that is dynamic and can be targeted and replaced. The brain cells themselves, once they're gone, they're gone. Very few can replace these. So you understand that the synapses are a very important thing to target because A, they store memories, and B, they keep the brain cells alive, the neurons alive. So we can use this graph, and I think this is the most important representation of what's happening in neurodegeneration that you should all try and remember. The common processes are that over time, and this is time, over time the number of synapses in the brain declines, and the amount of misfolded protein that occurs in all of these diseases, in Alzheimer's it's A beta and tau, in Parkinson's it's alpha-synuclein, in ALS it's TDP43, etc. But in all the diseases, there's an accumulation of misfolded protein over time and a decline in synapse number over time. And that happens, this happens anyway in aging brains, but it happens faster in neurodegenerative disease. And the reason this is so important is that it's very silent for a long time. So only when you've reached about 60% of loss of your synapses do the first memory problems come into play. And so that's when people come into clinic. They can't remember, they're being forgetful, they have problems with their movements, etc. You then just need to lose 15% more of your synapses, and you start to get neuronal loss. And this is the phase that you can intervene in, that you can recover, that you can treat, and this is the phase that is untreatable. This is the phase that leads to progressive loss of all your brain functions and cognition, institutionalization, and death. So you can see how important the synaptic phase is. The accumulation of misfolded proteins relates to both loss of synapses and brain cell loss, and I'll tell you how they all interact with each other. But you can all understand that imagine if this point here, which is first presentation, is when someone is 60, and this point is when they are 70, you can see that if you change the slope of that curve and raise this, then the point when brain cell loss occurs is so much later that it becomes irrelevant, and therefore you maintain function and health before you are institutionalized and have neurodegeneration. So the important thing is that if you have common mechanisms, you likely have common cures. So how do we study these mechanisms? I showed you a list of diseases. Mice, you can't do experiments in humans terribly easily. We can do them in mice. Mice don't get Alzheimer's disease, but they do get prion disease. So instead of being a sort of only partial copy of what's happening in the human, which with the Alzheimer's mice, prion disease mice have prion disease. They have biochemical, infectious, clinical, and pathological. They are identical to the human disease. So we use these mice to study the link between the aggregates of protein here and the dead neurons here. Let me see if I can get you a better pointer, actually. I think I can do a laser pointer. Yeah, here we go. Mind you, it's red. So here are the dead neurons. This is neurodegeneration in the hippocampus, the seat of memory. And here you can see all the dying synapses. So the prion disease mice let you understand why this protein misfolding relates to synapse loss and neuronal loss. So here I'm just showing you the basic representation of this protein misfolding. So all of these diseases have it. This is prion protein, but it could be A beta, it could be tau, it could be TDP43, etc. There's a native form, which is soluble, digestible by proteinase Ks and doesn't aggregate. and it converts with a change in its structure, and its tertiary structure, into an aggregated form. And this is the amyloid that you saw in the previous plaques, and this cannot be dissolved. Same primary sequence, same native, but a change in shape. And this is at the heart of all these disorders, this protein misfolding. And you can all understand this quite clearly by looking at this. Imagine this is a protein. This is exactly what proteins are like. This is correctly assembled to be functional. This is that same protein, aggregated, stacking up, non-functional. So the relationship between this and that is very much like the relationship between this and that. It's pretty much identical. As you can see, this is a non-functioning protein that's just accumulating. So this is at the center of all of these disorders. And what's interesting is that although the misfolded proteins have had so much attention because they are associated with disease, so in the Alzheimer's brain, all the plaques I showed you will be full of A beta or tau, the tangles. In a Parkinson's brain, they'll be full of synuclein. In Prion brain, they're full of this. But actually, all the treatments that attempted to reduce or disaggregate the amyloid have been pretty ineffective. So for my PhD, I asked a different question, because what's very clear is that if you don't have the native form, this does nothing. So it's the conversion of the two forms during disease that somehow is related to toxicity and cell death. So for my PhD, instead of tackling the PRPSC entity, I asked the simple question is, if you remove the native form for conversion, how does that affect disease? And what I did was I made mice. These are mice that express normal forms of prion disease, of prion protein. They all die within 12 weeks. But if you knock out the basic protein in these mice, at some stage during the disease, you completely cure them. So all of those mice, although they're infected with prions, will survive for their whole life. So you can see now that you've changed the concept of toxicity from this being the toxic entity to this process being the toxic process. And if you target this part of the process, you can save the mice. So this was a big story, and it's led to a change in policy with targeting the native protein rather than the misfolded protein for treating these disorders. But the thing that interested me very much about the mice that we depleted the prion protein in, here is a mouse with a healthy hippocampus, early in disease. Here are these spongiform vacuoles indicate synapse loss, but the neurons are still healthy. Just 4 weeks later, you can see how much neuronal loss there is. This whole hippocampus has shrunk. CA1 to 3 is gone. In the mice in which we depleted PRP, at this point in time, you reverse all the synaptic loss and you protect the neurons. So that's those mice that you saw surviving in that graph. And this is the neuroprotection. And we showed that first morphologically, then we showed that it's associated with recovery of memory and behavior. And finally, we showed that we could do it not just with double transgenic animals, but just using a virus targeting into the brain the native protein to deplete it. So that's a fairly robust indication that early neurodegeneration is reversible. And you can see that here. And what was very clear to me at that time was that if this is true for prion disease, this will be true for all neurodegenerative diseases, because neurons can only die in a certain way and synapses can only recover in a certain way. What my belief was that it doesn't really matter what the key protein is. It's the stress of the conversion and the toxicity that determine what's happening next. So this process, these mice, this process gives us access to key neurotoxic and neuroprotective pathways. And so the rest of this talk will be really to show you how we've used this model to discover pathways that could lead to treatments for dementia. So how do we understand them? We did a very simple analysis. So these are the mice that have had the PRP depletion at this point. So we do a very simple analysis of the earliest stages of disease to see what's changing. And the thing that was very consistent, remember that all the controls die at 12 weeks. So the thing that was very consistent was that about 2/3 of the way through their disease, the levels of all synaptic proteins are dropping. So that's about here. At this stage, forget about the treatments. Let's just look at the course of disease. The first thing that changes while the prion proteins are aggregating in the brain is that synaptic protein levels drop at about this stage. And that is associated with changes in memory and motivational behavior. This is a mouse that is doing a burrowing activity, which is a CA1 dependent activity. The mice excavate pellets from a tube, and I'll show you this later. This can be measured, and it's a behavioral quantitation of motivation and CA1 function. So that changes, and neuronal death follows. You'll see that at 9 weeks, the synaptic proteins go and the synapses start to go, but then you start to lose the neurons. So the critical point is the reduction in these synaptic proteins around here, and this is the point about how important synapses are for neuronal health and functioning and survival. So why does it happen? Let's go back to the misfolded proteins. This, you remember this accumulation of clumped up non-functional proteins? That's a stress for the cell. The cell needs correctly folded proteins to function. When they're like this, it's stressed. When they're like this, it's fine. And there's a very highly conserved signaling response called the unfolded protein response, or UPR, conserved throughout cell biology, or all, from yeast through to humans. And it's got 3 branches. I'm not going to focus on all of these, but the whole point of the UPR is to refold those proteins. So 2 branches upregulate target genes that will lead to chaperone expression, and 1 branch leads to, this is the PERK branch and the IF2 alpha branch, will reduce protein synthesis rates while the stress is resolved. So that's the branch I want to focus on. Here's the misfolded protein stress. As this accumulates, PERK is phosphorylated, EIF2 alpha is phosphorylated, and then you cannot put methionine on the beginning of a protein chain, so you can't make proteins. So that's a way of reducing the amount of production so that you reduce the production of the misfolded protein, and then the chaperones come in and refold. Very soon after this happens, this phosphatase is also induced, which then removes that phosphate group and restores protein production. So this is a standard homeostatic response, where that's the stress, you reduce protein synthesis, stress is alleviated, you restore protein synthesis. What we discovered was that during prion disease, as the misfolded prion protein accumulates, and this is a Western blot showing you the rising levels of that misfolded protein. So that's the plaques and the aggregates that you would see, but this is just on a brain slice, but this is on a Western blot. What we realized was that in parallel with this accumulation of misfolded protein, we were seeing a very marked increase in the levels of the IF2 alpha phosphate. And what you'll see here is actually an early response is increase, and then it is reduced, but then suddenly it fails to be reduced, and you get a really exponential and prolonged increase in this. I can't emphasize how much this is unphysiological, because the normal response I showed you, this happens in minutes. You switch off protein synthesis, and within minutes it's restored. So here, this 2 alpha level is sustained for weeks, not minutes, in old brain cells. And what that translates to is a reduction in protein synthesis rates in neurons that are diseased, which is also lasting for weeks. So our hypothesis was that actually synapses are lost and brain cells are dying, not because of any specific toxic effect of the protein accumulation, but because of overactivation of this pathway. So, it's saying that this dysregulation is causing a critical reduction in essential proteins that leads to neurodegeneration. So we had to prove this, and to prove this, we basically are trying to target this phosphate group. We want to reduce the levels of EIF2 alpha in the context of disease. So one approach is to do RNAi of the actual misfolded protein so that none of this is activated. Another approach is to put extra phosphatase in so that you remove this and lower the levels of 2-alpha. And then, of course, the control that you need is something that blocks the dephosphorylation of 2-alpha. So here's the experiment. Control mice. Mice infected with prions only. Remember, these will die in 12 weeks. They'll lose their neurons from 10 weeks, and the UPR is superactivated from 9. And all of these other mice are being treated to affect the 2-alpha phospho levels. And the ones I want you to concentrate are these two, because these are the two interventions that are going to reduce the levels of EIF2-alpha phosphate. So let me show you a Western blot showing how the chemistry works. So here's the levels of 2-alpha phosphate in the prion-only mice. And here you can see if you give the virus with GAD34, they've dephosphorylated 2-alpha. If you give RNAi in the disease, you prevent the phosphorylation of 2-alpha. And if you give salubrinol, it's worse. And that's what, so all these things are biochemically acting as they should. What's the effect? Protein synthesis rates come up, as you would expect, because you've got lower levels of EIF2-alpha. You've got protein synthesis rates restored. This restores synaptic function. It restores burrowing behavior, which I was telling you about. It restores synapse number. You can see that this is returned. These are electron micrographs of synapses. These are mice with prion disease. These are control animals with very low synapse number, and these are the ones treated with the dephosphorylating 2-alpha. And above all, it's unbelievably neuroprotective. Plus, it has a very significant effect on survival. And these animals just had a single injection of these viruses into the hippocampus and yet got very highly significantly increased survival. So we proved our hypothesis in that if you can target levels of 2-alpha phosphorylation in the context of disease and restore translation, you get neuroprotection. So this was very exciting. And I put this blue line here because it doesn't matter what your upstream protein is, if that's the case. If you target this process, this could be potentially, it could be A beta, it could be tau, it could be prion, it could be anything you want. And that was a very nice story, and we were very pleased with it. But it was genetic proof of principle, where you show the pathways important in a molecular way. But really, for this to have any impact in the clinic, you've got to be able to show it pharmacologically. So can we do it with drugs? So if you look at the pathway again, this compound here was generated to inhibit PERK, and so prevent the phosphorylation of 2-alpha in cancer. And GSK had made this, and it wasn't a very good cancer drug, so they shelved it. So they gave me some, and we basically tested this molecule now in the prion model. So here they are, the mice being inoculated. Remember, they die here. Remember that synapse loss starts at 7 weeks, and the kind of protein synthesis and behavioral deficits at 9, and then neuronal loss. So we're now treating orally with this drug from clinically relevant moments, not from the very beginning of disease, but when they're actually starting to have significant disease. And I'm going to show you the effects here. So you're supposed to show. No, hang on. Sorry. Where's my... I'm supposed to play me a video. Why isn't it playing? Where's my videos gone? Oh, what a shame. Let me see if the next one works. Sorry, guys. This should be... We should have checked this, Mosab. Right. Hold on with me a minute, because I'm going to show you something, if I can. I might just switch to another presentation. Just give me 1 second. Is that going to work? Is it going to work? Can someone speak to me?
I don't think it's working.
No, the videos aren't working. Let me see if I can share another presentation. Can I do that for a second?
Sure.
Okay, sorry guys. Sorry the world. I'm just going to have to exit for a second. I guess I could just carry on without it. It's a shame, though. It'd be nice to show you the videos. It's very impressive. What do you want me to do? Oh, here it is.
I think it's,
gotcha.
yeah, fantastic. Okay.
Right, guys, so we're back on. These mice have both had the same amount of disease. This one's had the drug. You can see that the one on top really cannot move. He's very unsteady. His tail's in a very weird position. He's about to fall over. Look, there he goes. Whereas his litter mate, who's got exactly the same amount of toxic protein in his brain, is perfectly fine. Let me see if I can do you another one. Yes, they come back up. Brilliant. Here's one, a treated mouse. The untreated mouse has got paralyzed hind limbs. His litter mate, which has got the drug, is perfectly fine. And here, let me show you this one. Let me see if this activates. Yes. This animal is doing what it's supposed to do. He's been treated, and this animal is absolutely failing to do a really important motivational task. He's got no burrowing activity at all. That means his CA1 in his hippocampus is not working. The drug-treated animal is cured. Let's just show you those again. The hind limb paralysis, the general posture. You can see the total cure of the treated animal. And right at the end of disease, no healthy animal can be put on its back. See the one underneath? That just gets right on it. This animal is so neurologically impaired that it cannot turn itself over. It has no reflexes. It's just unable to move properly. So this is a really, really important discovery. This is clinical cure of neurodegeneration, of a neurodegenerative disease, of a really rapidly lethal neurodegeneration. Neurodegenerative disease with a medicine. And how's it doing it? It's restoring protein synthesis rates, and look at the effect on the brain of the drug. It completely protected hippocampus compared to vehicle treatment. So this was the first small molecule to treat neurodegenerative disease, targeting a generic pathway. And it was a really very big news story. And it was hailed by the BBC as a breakthrough in Alzheimer's disease, even though we'd done this in Prion disease mice. And this is because the BBC were aware, as were we, that actually this pathway is activated all of these diseases. So Eric Klan's lab in New York showed at the same time as we published our paper that 2-alpha kinases, suppression of other 2-alpha kinases like PERK alleviated Alzheimer's deficit. Claudio Hetz used the same small molecule to cure a Parkinson's disease model, and Nancy Bonini to cure an ALS model. And the brains of human patients are full of phospho-PERK and phospho-2-alpha in Alzheimer's disease, in other tauopathies like supranuclear palsy, in Parkinson's disease, and in ALS. So the relevance of the pathway is really well established in multiple labs around the world. And we finally found a dementia model mouse. This is P301L mouse. This is a frontotemporal dementia mouse. I don't have a video here, but you'll see how unwell that is and how thin its hippocampus is. And that's the effect of treating with the drug. So that's so several mouse models, same pathway, same effect. And that's what you really want to see when you're proposing a common mechanism. But the but is this, that the GSK molecule, whilst protecting the brain, poisons the pancreas. So this is pancreatic necrosis. And of course, you can't give a drug that does this to humans. And it's doing this because this is such an efficient inhibitor of the UPR, of the PERK branch, that a cell like the pancreas, which needs to make enzymes and needs to switch off its protein synthesis rate, will die if you give it this drug. So we were not deterred, because what I didn't tell you about as well is that protein synthesis at the synapse is essential for memory. And there's a great deal of work been done on phospho-2-alpha levels and memory. And Peter Walter and Noam Sundberg made this molecule, which inhibits the effects of VI2-alpha phosphorylation, not here, like this molecule, but downstream. And what they had found in this paper was that ISRIB, by restoring protein synthesis, made wild-type mice smarter. So it made them make more proteins at the synapse, and they had better memory. So we made the molecule and we tested it in prion mice. And here's the effect of the GSK molecule, and here's ISRIB. So pretty much the same degree of neuroprotection, but critically, here's the pancreas, and the pancreas is saved. So ISRIB is of key importance because it shows you that you can inhibit the UPR safely without damaging the pancreas, which was the great fear around molecules like GSK. And it does it because it does it partially. So here's the inhibitory effect of GSK, and here's the effect of ISRIB. So the partial restoration of protein synthesis rates allows the brain, protects the brain and the pancreas. So there is massive drug discovery efforts around this now. There's millions of patents on ISRIB and ISRIB-like molecules in dementia, in head injury, in all sorts of in cognitive enhancement. But ISRIB is not suitable for humans because it's highly insoluble and it's a very difficult molecule to get into the clinic. But it's a big focus for drug discovery. So there are two ways that you can find drugs to do what you want. One is you can design them through drug discovery programs, which takes years, if not decades, and a lot of money. Or you can do a repurposing screen. And here you use a library of largely known drugs and here my student Mark, he's now a postdoc, but he was a student in the lab, used a library of drugs where he found this is ISRIB, this is GSK, and he was looking for any drugs that do the same to the UPR, and he's found these two. And just to cut a long story short, these two, this is licensed, this is not, but it's nearly licensed. They're both as neuroprotective, they're safe in humans, they get into the brain, they treat both prion and frontotemporal dementia mice, and they increase survival. And this is a way of measuring the effects of trazodone on protein synthesis in brain slices. You'll see here, this is wild type brain with protein synthesis. Every new protein is labeled green in the cell body, in the synapses, in the dendrites, in the neuropil. In prion disease, you can see how devastating the effect is on synaptic protein synthesis. And with trazodone of 2 weeks treatment, you can see that it restores protein synthesis. So this is now ready for. We're going to do two things. We're going to measure protein synthesis rates in human patients, and this is the first brain scan of everything I've been showing you in mice so far. This is a human control patient in whom we're measuring protein synthesis in vivo in the brain, and the highest is white and the lowest is red or black. We're about to be doing patients for that, so we're going to ask the question of whether trazodone can do this in patient brains, because then it really would be a very exciting candidate for clinical trials in dementia and Alzheimer's. That's the first half of my lecture. So I've shown you how the common pathways engaged by protein misfolding can be manipulated, A, to save synapse number, and B, to save neuronal loss. But there's another. If you didn't have this loss in synapses to start with, none of this might matter. It may make much less impact. So the question is, why are synapses dying? So here is, your synapse, which is so trophic and important for the neurons. Here it is under EM. I want you to just take a good look at it. Vesicles, cleft, and PSD. Vesicles, cleft, PSD. That's what makes a synapse. The loss of synapses happens early in neurodegeneration. Whichever disease you choose, this is the part where the UPR is activated and symptoms start, but synapse loss starts way before. So there's this long silent phase when you've got lost nearly half your synapses. So the real question is, why is this happening? Now look, synapses are constantly being made and dismantled by a balance between pruning and regeneration. So why is that happening? Obviously, that balance is off. You can have more pruning, and there's evidence for increased microglial pruning, or you could have reduced regeneration. And our hypothesis was that synapse regeneration is deficient in neurodegenerative disease, as well as, or it's separately to pruning, but we had a distinct hypothesis that this is what's going wrong. So how do we test this hypothesis? So as I said, we're all continuously making and regenerating synapses. And there are ways of putting a window onto the cortex and seeing synapse plasticity as it happens in vivo, but that's very high tech. So we went to an area of biology where we know that structural plasticity is happening very vividly, and that is hibernation. Now, this is a hibernating dormouse or ground squirrel, in fact. And what happens during hibernation is you reduce your body temperature right down, you reduce your metabolism right down, you reduce protein synthesis rates to save energy, and you dismantle your synapses. And this is not our discovery, this is well known. So here's a normal synapse, which I was showing you before, synapses cleft and PSD. Here's a cooled synapse. And what you'll notice is the PSD has gone. And so the vesicles pull because there's no transmission. And on rewarming, this comes back. So this is a phenomenon in biology by which you can save energy. And it occurred to me when I first learned about this phenomenon that whatever happens in hibernation also happens on a much lower scale during sleep and during normal repair and plasticity. So if this process is at all defective, you can imagine that you will, if you're going through the cycle of synapse dismantling and reassembly, if you can't reassemble properly, then you will lose synapses. So the first thing we did was to prove that this could happen in wild-type mice. So here's a wild-type mouse. Wild-type mice are not hibernators. I've pseudo-colored the synapses so you can see them more easily, where the post compartment is green and the pre is yellow. Cooling, you can see, reduces the number of synapses, and rewarming restores them. So although this is our control, this is a very, very important phenomenon because this shows you that this is highly conserved in nature, and it's not a phenomenon just restricted to the hibernators. And you can count and quantify the synapses in a very significant increase. So we then tested disease models, and here's our Prion mice. And now we can use some of the other mouse models because these do have synapse loss, even if they don't have neuronal loss. So we're testing before, remember that synapse loss happens here. So what you have to ask is the capacity for regeneration healthy or impaired before you start to lose synapses? So we took the disease models and we cooled them very early. Now, you might remember, I hope you remember, that in prion disease, this starts to happen at 7 weeks. So we have to cool them before 7 weeks. So if you cool them at 4 weeks after infection, you lose and regain synapses normally. So that's healthy. But if you cool them at 6 weeks after infection, you fail to regenerate your synapses on rewarming. So our hypothesis is correct in the prion model. In the Alzheimer's models, they lose their synapses from 4 months. And at 2 months, they're plastic, cool, you lose, rewarm, they come back. But at 3 months, you cool them and they can't remake synapses. So you can see that there's a loss of synaptic plasticity, structural plasticity that is preceding synapse loss. And this is the earliest deficit in regenerative capacity, preceding everything else. And you can see that if you could fix that, then you might delay or even prevent all of the next stages. What's the mechanism? So I told you that this is mediated by cooling. And cooling is famous for being neuroprotective, and hypothermia is used medically, as I will tell you later. And they induce the downregulation of protein synthesis metabolism, except for a group of proteins called the cold shock proteins. And these are upregulated. And these are RNA chaperones. And what they do, if you imagine that in hibernation, if you're shutting down protein synthesis, when the mouse wakes up, or the squirrel, or whatever it is, wakes up, it needs to start translation again quickly. And so the RNA chaperones have a host of RNAs ready for translation. One of these is called RBM, RNA binding motif 3, RBM3. It's highly expressed in brain. known to be expressed in hibernation in various species. And here it is in our mouse brains on cooling. So we're finding it in wild-type mice. And we discovered that the failure of synapse regeneration coincides with the failure of induction of RBM3. So here it's induced. Remember, 4 weeks in prion disease, the synapses come back. At 6 weeks, they don't. That's associated with low levels of RBM3. In the Alzheimer's mice, at 2 months, the synapses come back. At 3 months, they don't. And again, the RBM3 is low. So is this just an association, or is it actually causal? So if we increase RBM3 levels, can we restore synapse regeneration? So we did this in 2 ways. We first cooled the mice before they lose the regenerative capacity and before they lose the capacity to induce RBM3, which happens at 6 weeks. So we cool them twice, and then we follow what happens. And these mice aren't cooled. And I just show you that if you cool the mice, the RBM3 stays up for weeks and weeks, which is what you'd expect for hibernation. And the early cooling prevents synapse loss in prion disease. So normally at this stage, you would see this much synapses or less, but the cooled mice have got the same level of synapses as wild-type mice, so they're entirely protected. And what's the effect of that? It prevents behavioral deficits that you've seen, the burrowing. It prevents the loss of memory. This is novel object memory. And look at the effect on neuroprotection. These mice haven't had any drugs. They haven't had GSK. They haven't had ISRIB. They haven't had trazodone. They haven't had anything. They've just been cooled. And look at the degree of neuroprotection. It's really extraordinary. And the effect on survival was also extraordinary. This outlier shows you just how protective cooling can be. So can we mimic the effects? Because of course, cooling will do more than just put up RBM3. So how much is RBM3 the main mediator of this? So now we go back to our lentiviral approach. And here we're going to overexpress RBM3. And here we're going to knock it out. And now we don't cool the mice. We just give them prion disease. And here's a prion disease at 9-week control and Alzheimer's mouse at 6 months when they've got established synapse loss. And here are animals that were treated with RBM3, and you can see that that has protected their synapses. The same is true with behavior and memory. The same is true with neuroprotection. And that's the effect of RNAi. If you block the RBM3, you completely it's even worse than the controls. So you can see that almost all of the work of cooling is being done by RBM3. And again, it increases survival. So this is the pathway. It's more complex than that, but I'll show you that in a minute. And this again was a very nice story that the BBC latched onto, saying that hibernation hinted at dementia therapy. They very conveniently showed us bears. So I showed you mice, and mice are small mammals, as are squirrels, dormice, and many hibernating animals. And the difference between large animals, and this bear, I think you'll agree, is a large animal, and small animals is that bears hibernate at 35 degrees. So that's a temperature that you could entertain and cooling people to, and that is the temperature that they use in therapeutic hypothermia. Whereas we were cooling those mice to 16 degrees, which is obviously not feasible for humans. So the idea is, obviously we can't cool everyone. It's not practical. It's also not good for you. It gives you pneumonia, it gives you blood clots, etc. So the idea is to be able to induce RBM3 without cooling. That would be the therapeutic approach. But for this, we need to know what the mediators of RBM3, what controls RBM3 without cooling, so how it's induced, and the relevance for human disease. So what are the, so here are the mediators. The main mediator is this protein here, RTN3, which is a reticulin protein. And that, I'm not going to show you all the data, but that does everything RBM3 does. So if you knock out RTN3, you can't neuroprotect. So RTN3 is the main effector of RBM3. What is the pathway? Well, the pathway is recently been described by Diego. It's a non-canonical pathway where TrkB is activated through a specific pathway that induces the neuroprotection and completely orchestrates that synaptic remodeling. But what this tells you is that this is druggable. So we can we can induce RBM3 without having to cool. And the relevance for human disease is in the fact that we use therapeutic hypothermia. Whoops. This baby is being cooled, but adults with head injury, patients having cardiac surgery are all cooled. And this baby's been cooled to 35 degrees. How cooling works is not understood. These people voluntarily get hypothermia. They swim in frozen water, and we've been working with these people. And we've tested RBM3 in human brain from patients having surgery, and you will see that it does go up. So RBM3 is a relevant target in humans. It goes up in the babies, and it goes up in the swimmers. So the importance here is that we do have a cold shock response in humans that is like that in mice. It can be drugged. And here you'll see Alzheimer's brains where the levels of RBM3 are higher in controls than they are in Alzheimer's disease. So it's also relevant in disease. So I'm going to finish there, but I hope I've shown you two separate pathways, common pathways that are independent of specific disease, where either through UPR activation here or through failure of structural plasticity here, they contribute to both of these processes, to synapse decline and neuronal loss. And that if we intervene on either side, you can see that we change, we stop the decline of synapses and the neuronal loss is deferred. And if you do this for patients, you will keep them with their memories and out of institution and without the devastating progressive phase of neurodegeneration. And one of science's unanswered questions is, to what extent can we stave off Alzheimer's and Parkinson's? And I think a 5 to 10 year delay in this late onset disease would improve old age for millions. And I think if we can do what we've done in humans as we've done in mice, that this is totally achievable. So I think that's where I want to leave it. Okay, I finished.
Fantastic. Thank you very much for a fantastic presentation. So we're going to take some questions now, and I'm going to be selfish to ask the first question.
Do you want to see me, Mosab, or?
No.
That's fine. I'm happy to be invisible.
Okay. All right. So let's do this. Okay. Yeah.
Yeah.
So my question is, do we have like any evidence, for people, for example, living in the cold areas who have the tendency of developing a familial form of either AD or something? Do we have any evidence that they have like the age of onset becomes a little bit delayed due to this?
So I can understand that question, but there are two things to remind you about. The people who live in the cold areas have the same body temperatures as the people who live in Africa, because we're homeotherms. So they are not colder than we are. Nor do they get cold if they can help it. Only those crazy people get cold, the ones I showed you. So they don't. But the other thing about cold areas is that they tend to have very low life expectancy for other reasons. There's diabetes, there's lots of alcoholism, there's suicide and early death. You don't have an elderly population that you can assess. But above all, people who live in cold climates keep warm. They don't do what those crazy swimmers do, which is get cold.
Yeah, makes sense. Okay, so I'm gonna just randomly select questions that are relevant to the presentation. So there is this person. He said, is it feasible to study Alzheimer's disease in mice? And I guess this is probably because at the beginning of your talk, you showed this Prion model that they developed Prion disease in mice, while in AD, probably it's not straightforward.
So there's no good mouse models of Alzheimer's. So the best we can do is study the effects of protein misfolding on neuronal health, because mice don't get Alzheimer's disease. There's been a lot of modeling that you can make mice that will make plaque, A-beta plaque, but you don't get mice that will do what happens in human disease. And that's partly due to the time, the duration, and the whole natural history of Alzheimer's, which is generally a disease of old age, which comes late after decades of protein misfolding and cell stress, whereas a mouse model is inevitably crammed into a year or two. So you can't feasibly, you can model aspects of the disease, you can model common processes. And in the end, you know, even if you could, you still have to do the experiments in humans. You still have to take patients and do experimental medicine studies and clinical trials.
Absolutely, absolutely. So we've got this question. I'm not sure what it is asking, but it says, being in the tropics, Nigeria, so the person is from Nigeria, how will cooling difference alter synaptic associations in the hippocampus during hyperparasitemia, which lead to cerebral malaria compared to, say, wild-type mice?
I didn't understand the question. Sorry.
Yes. So the person is basically...
Are they asking about hyperthermia, about fever?
I think so. So how will cooling difference alter synaptic association in the hippocampus during hyperparasitemia? So they're talking about hyperparasitemia here, which leads to cerebral malaria compared to, say, wild-type mice.
I think cooling's always going to be protective, whatever else is going on. And I think hyperparasitemia is often associated with a fever, which is also not good for you. But I mean, it's a generic protective mechanism, and that's really all I can say.
Okay, so we've got another question here. The person said, "Thanks for the exciting talk. Talking about inducing RBM3 with cold, is RBM3 produced in the brain by the neurons or from other non-neuronal tissues within or outside the brain?"
Good question. Definitely neurons, probably astrocytes. Definitely astrocytes in babies. Definitely neurons in adults. Probably both. But it's a good question.
Oh, that's really good. So before we have the next question, I was just thinking, what do you think we need to, because there are a lot of early career researchers listening to this talk, what do you think we need to focus on to succeed as early career researchers?
Well, you all need to develop your own area, really, of expertise, and that's very difficult when you're early career research. But really, that is the secret of success: is finding your own niche, however small. Mine was focusing on the reversibility of prion disease. That allowed me to develop that was my unique territory, which has allowed me to exploit and discover these pathways. So I would really encourage you to find what you're very interested in and try and develop a story that becomes your own territory within it. Trying to compete with established giant labs, if you all take up autophagy tomorrow, that would be hopeless, because there are huge labs all around the world. So try and find a niche area that interests you and that you can develop in your own way.
And hopefully you get funded.
Yeah, I know, it's very difficult.
Okay, so we've got a question from our boss, the Secretary General of the Society of Neuroscientists of Africa, Professor Amadi Inhuo. He said, in quantitative terms, have you estimated the extent of recovery from the neuronal loss after the reworm process?
Yeah, I mean, we've quantified it, absolutely. I mean, it's almost recovers to normal. Yeah, we've got, we count the neurons in slides using new N staining, and so yes, we have quantified that.
Okay, thank you. So before we have the next question, I was also thinking, I mean, I've followed your work for a long time, and it's always just kind of fascinating and inspiring to see the success of the work that you do. So what inspires your work? What inspires you? For example, you were talking about this different pathway, then you move to cooling. I'm just kind of interested in knowing what inspires you to do this amazing work.
I mean, I'm just interested in biology and what how cells respond to stress and how they fix themselves. And both of these processes are as simple as that. If any biological process is telling you something, And the thing that we see in these diseases is synapse number go down. So my first thing is, so why is that happening? Just ask a question. Why is it happening? That's the interest that opens a door. And then it was seeing the lecture on hibernation, which I saw when I realized that the synapses were coming and going away. For me, that meant that this must be a conserved process, and it's probably going wrong in these disorders. So that gives you a hypothesis to test. We tested it and we were right. And that's really fantastic fun in science when you can do that, when you can really come up with a question, test it, it's right, and then it allows you to go on. The UPR story was similar. I knew the UPR was activated all over in all these diseases. So I just tested for it, saw what happened to protein synthesis rates, and it's obvious if you deprive cells of neurons of proteins, or any cells of protein, for long enough, they'll die. And if you just fix that, then that recovers. So it's the satisfaction of testing simple biology and truths, really.
I see. That is really fascinating. So we've got this question. The person said, are organs like the brown adipose tissue and liver important for regulating metabolism, implicated in communication with the brain? And if they can also serve as a source of RBM3?
That's a great question. I have no idea whether they have RBM3. We've never looked. But they probably do signal in a paracrine way. But again, I just don't know that. I think that the whole metabolism is so intricately linked that I think they must signal and get wake the brain up again and make the synapses come back together. But I don't know the answer.
I see. So I was thinking, before we get another question that's relevant, so because based on your work, definitely you can rescue neurodegeneration, even though we have misfolded proteins. I was thinking, therefore, with all these failures in their clinical trials for Alzheimer's, especially focusing on A beta etc., do you think, therefore, maybe focusing on downstream pathways is beta instead of focusing on, say, amyloid beta, tau, etc.?
I think the data speaks for itself. There have been about 450 failed trials of A beta immunotherapy. That doesn't mean that A beta's a hopeless target, but it's the difficulties with targeting it at the right time in the correct group are very clear. And I think it's time for new ideas, new directions. I think if you target common processes, what you do is you don't target the root. It's like targeting A beta is like, to some extent, a simple way of looking at it is, imagine you have someone who gets a chest infection or pneumonia, and then they develop sepsis. So that's multiple failure. When they come back to you, treating the pneumonia won't cure them. You've got to maintain all the vital functions. And that's a little bit what I'm proposing here, that you've got to give neurons back their protein synthesis. At that stage, the misfolding protein isn't the problem. It's the dysregulated stress responses that are really killing the cell. So you've got to target what's actually wrong with the cell at that stage, not what the original cause is. So it is the original insult, but actually in a demented patient, they've got a brain that's out of control.
Fantastic. So we're going to take these last two questions. So the first one said, can we consider neurodegeneration as infectious disease? I think that's basically what the person is trying to say.
No. I think some, Prion disease is an infectious disease. And there is evidence for propagation of misfolded protein, but whether that is infection is another matter. So I don't think they're not infectious diseases in the way that malaria or streptococcal infection is. No, but Prion disease is an infectious disease, that's for sure.
Okay, and the last question. So he said, "Thank you for educated presentation. Does it mean that a potential drug candidate in Prion disease can act against Alzheimer's?"
Yes, that's exactly what it means.
Absolutely.
Against Alzheimer's, against Parkinson's, against motor neuron disease, yes, because these pathways are relevant to all of those disorders.
Okay, fantastic. I think we've run out of time. Okay. So on behalf of the Secretary General of the Society of Neuroscientists of Africa, Professor Amadi, SONA members, and organizing team of this webinar, I would like to thank you for being with us today and for this fantastic presentation. And I hope that all the listeners have taken some message, at least inspiration, from this amazing work that you're doing. Thank you so much, Giovanna.
Thank you very much for having me. Bye, everybody. Bye.
Bye.